Fundamental processes. Enzymes involved in DNA replication. CSIR-NET JRF Life Sciences level, from base to advanced. DNA replication is a fundamental biological process essential for the inheritance of genetic information during cell division. This process is tightly regulated and highly coordinated, involving a suite of enzymes that ensure accurate and efficient synthesis of a new DNA strand. The core objective of DNA replication is to produce two identical copies of a DNA molecule from the parent DNA strand, ensuring genetic fidelity. The entire process follows a semi-conservative model, where each daughter DNA receives one parental and one newly synthesized strand. 1. Helicase, DNA unwinding. Function. Helicases are responsible for unwinding the double-stranded DNA into single strands by breaking the hydrogen bonds between base pairs at the replication fork. In prokaryotes, the helicase is DNA-AB. In eukaryotes, the helicase complex is part of the MCM, mini-chromosome maintenance complex, composed of MCM2 to 7 proteins, and is loaded at the origin by CDC6 and CDT1 with help from ORC. Mechanism. These enzymes utilize ATP hydrolysis to catalyze the unwinding of DNA. Helicase activity generates supercoiling ahead of the fork, which is resolved by topoisomerases. 2. Single-stranded binding proteins, SSB, RPA. Function. Once the DNA is unwound, single-stranded DNA binding proteins, SSBs, stabilize the exposed strands to prevent reannealing and protect them from nucleases. SSB in prokaryotes. RPA, replication protein A, in eukaryotes. These proteins play a crucial role in maintaining the single-stranded structure necessary for DNA polymerase function. 3. Topoisomerase, relieving supercoiling. Function. As helicase unwinds DNA, it introduces positive supercoils ahead of the fork. Topoisomerases alleviate this torsional stress. Type 1 topoisomerase cuts one strand of DNA to relieve tension and then religates it. Type 2 topoisomerase DNA gyrase in prokaryotes cuts both strands and passes another part of the duplex through the break to relieve supercoiling. Topoisomerases are essential for allowing fork progression and are targets of antibiotics, e.g., quinolones, and anti-cancer drugs, e.g., doxorubicin. 4. Primase, synthesis of RNA primer. Function. DNA polymerases cannot initiate synthesis de novo and require a free 3'O group. This is provided by short RNA primer synthesized by primase. In prokaryotes, the primase is part of the DNA G protein, which associates with DNA B helicase to form the chromosome. In eukaryotes, DNA polymerase alpha has primase activity, synthesizing a short RNA DNA hybrid primer, approximately 10 RNA plus approximately 20 DNA nucleotides. Primers are removed and replaced with DNA by specialized polymerases. 5. DNA polymerases, core of replication. Function. Catalyze the addition of nucleotides to the grown DNA strand using the template strand, always in the 5' to 3' direction. Of prokaryotes. DNA polymerase 3, main replicative enzyme, with high processivity and 3' right pointing arrow 5, exonuclease activity proofreading. DNA polymerase I, removes RNA primers using its 5' right pointing arrow 3, exonuclease activity and fills in the gaps. DNA polymerase 2, IB, B, involved in repair and translation synthesis. B, eukaryotes. DNA polymerase alpha alpha initiates DNA synthesis after primase action. DNA polymerase delta delta synthesizes the lagging strand, has proofreading function. DNA polymerase epsilon epsilon synthesizes the leading strand, also possesses proofreading activity. DNA polymerase beta and gamma, involved in DNA repair and mitochondrial DNA replication, respectively. 6. RNase H and FEN1 primer removal in eukaryotes after replication. The RNA primers are removed by RNase H, removes most of the RNA primer. FEN1, flap endonuclease 1, removes the flap structure created by polymerase delta. These are replaced by DNA and sealed by DNA ligase. 7. DNA ligase, joining DNA fragments. Function. Seals the mix between Okazaki fragments on the lagging strand by catalyzing the formation of a phosphodiester bond between adjacent nucleotides. Uses ATP in eukaryotes and NAD+ in prokaryotes. Essential for genome integrity and proper chromosomal replication. 8. Sliding clamp and clamp loader, enhancing processivity sliding clamp. Beta clamp in prokaryotes. PCNA, proliferating cell nuclear antigen in eukaryotes. It encircles DNA and tethers DNA polymerase to the template, enhancing processivity. Clamp loader. Gamma complex in prokaryotes. Replication factor C, RFC, in eukaryotes. These load the sliding clamp onto DNA at the primer template junction. 9. Telomerase, extending chromosome ends. Function. In eukaryotes, linear chromosomes have the problem of end replication. Telomerase, a reverse transcriptase enzyme, adds telomeric repeats to the 3' ends using its intrinsic RNA template. Active in germ cells, stem cells, and cancer cells. Inactive in most somatic cells, leading to progressive shortening of telomeres during aging. 10. DNA proofreading and error correction 3' to 5' exonuclease activity. Performed by DNA polymerases 3, delta, and epsilon. Removes misincorporated nucleotides immediately after synthesis. Mismatch repair enzymes correct errors that escape proofreading, ensuring fidelity of DNA replication. Previous year CSIR-NET question CSIR-NET June 2020. Which of the following enzymes possesses both 5' to 3' polymerase and 3' to 5' exonuclease activity in prokaryotes? Of DNA polymerase I. B. DNA polymerase 2 C. DNA polymerase 3 D. All of the above answer. D. All of the above. CSIR-NET Deck 2018. The RNA primer in eukaryotic DNA replication is synthesized by a DNA polymerase alpha. B. DNA polymerase beta C. DNA polymerase delta D. Primase only answer. A DNA polymerase alpha. CSIR-NET June 2021. Which of the following enzymes is responsible for relieving torsional stress during replication? A helicase. B. Topoisomerase C. Ligase D. Primase. Answer. B. Topoisomerase. Final thoughts. Understanding the enzymatic machinery of DNA replication is essential for mastering molecular biology and is a core focus of the CSIR-NET Life Sciences syllabus. This topic integrates deep knowledge of protein DNA interactions, cell cycle control, and genetic fidelity. Special attention should be paid to the functional coordination between these enzymes, their regulatory mechanisms, and how these processes differ in prokaryotes and eukaryotes. Mastery of these enzymes also provides the groundwork for understanding DNA repair, mutagenesis, replication stress, and cancer biology, making it a repeated and crucial question area in the CSIR-NET examination. You said, replication origin ChatGPT said. Fundamental processes, replication origin. CSIR-NET JRF Life Sciences level, base to advanced maximum detail, paragraph form, no tables. The origin of replication ORI is a specific nucleotide sequence in the genome where DNA replication begins. It serves as the initiation point for the unwinding of DNA and the assembly of the replication machinery. Origins are essential for ensuring that the entire genome is copied precisely once during the cell cycle. The properties and number of replication origins vary significantly between prokaryotes and eukaryotes, reflecting differences in genome size, complexity, and chromatin structure. Prokaryotic origin of replication ORIC. In prokaryotes, such as Escherichia coli, the genome is circular and contains a single origin of replication called ORIC. This region is approximately 245 base pairs in length and is rich in AT, adenine, thymine base pairs, which are easier to unwind due to fewer hydrogen bonds. ORIC contains three key types of sequence elements: the DNA A boxes, which are specific binding sites for the initiator protein DNA A, an AT-rich region where strand separation begins, and other accessory sequences that help regulate initiation. Replication begins when multiple DNA A proteins bind cooperatively to the DNA A boxes in ORIC, causing the adjacent AT-rich region to melt and form an open complex. This is followed by the recruitment of the helicase DNA B and primase DNA G to form the chromosome. Helicase unwinds the DNA bidirectionally, establishing two replication forks, which proceed around the circular chromosome in opposite directions until they meet at the termination region, tear site. Only one round of replication is allowed per cell cycle, and this is tightly regulated by methylation via DAM methylase and the binding status of regulatory proteins. Eukaryotic origins of replication. In eukaryotic cells, the situation is more complex due to the linear and much larger size of the chromosomes. To ensure complete and timely replication, multiple origins of replication are per chromosome. Each eukaryotic chromosome can have thousands of potential origins, although only a subset is activated during each S phase. These origins are selected based on the chromatin state, DNA sequence, histone modifications, and replication timing program of the cell. Unlike prokaryotic ORIC, eukaryotic replication origins do not have a highly conserved consensus sequence across all species. However, in Saccharomyces cerevisiae, budding yeast, a well-defined autonomously replicating sequence, Rs of about 100 to 200 BP acts as the origin. Rs contains the A element, a consensus sequence required for binding of the origin recognition complex, ORC, and the elements that enhance ORC binding and helicase loading. The ORC origin recognition complex is a 6-subunit protein complex that binds to replication origins throughout the cell cycle. In G1 phase, CDC6 and CDT1 assist ORC in loading the MCM2 to 7 helicase onto DNA in a step called origin licensing. This forms the pre-replication complex, pre-RC. Upon entry into S phase, cyclin-dependent kinesis CDKs and DBF4-dependent kinesis DDKs phosphorylate components of the pre-RC, triggering origin firing and the recruitment of additional proteins such as CDC45 and GINS to form the CMG helicase complex, which unwinds DNA for replication. Once an origin fires, it is inactivated for the remainder of the cell cycle to prevent replication. This is enforced by CDK activity, which inhibits relicensing until mitosis is complete and CDK levels fall. In multicellular eukaryotes, origin selection and activation are further modulated by epigenetic marks and replication timing domains, which correlate with gene expression and chromosomal architecture. Regulation of origin firing and temporal control. Eukaryotic replication origins do not all fire simultaneously. Instead, replication is temporally regulated in a highly reproducible pattern known as the replication timing program. Some origins fire early in S phase, often those in transcriptionally active euchromatin, while others fire late, often in heterochromatin. The rate of replication fork movement, origin spacing, and replication timing are coordinated to ensure complete genome duplication. Checkpoints, particularly the intra-S phase checkpoint, monitor the integrity of origin firing and fork progression. In response to DNA damage or replication stress, checkpoint kinases such as ATR in eukaryotes suppress the firing of late origins and stabilize stalled forks to preserve genome stability. Comparison and functional significance. While the mechanistic core of replication, helicase loading, priming, polymerization, is conserved across domains, the regulatory architecture of origin usage has merged. Prokaryotic replication is rapid and simple, suitable for high-speed cell division. Eukaryotic replication, with multiple origins and tight temporal spatial regulation, is adapted for the complexity of chromatin and the need for replication fidelity over long genomes. Origin selection and regulation are also closely tied to developmental signals, cell type, and environmental conditions, highlighting their central role in cellular function and genome maintenance. CSIR-NET previous year question CSIR-NET Deck 2017. In E. coli, the initiation of DNA replication at ORIC is regulated by the methylation status of DNA. Which enzyme is responsible for this methylation? A. DNA ligase. B. DAM methylase C. DCM methylase D. DNA polymerase I. Answer: B. DAM methylase. CSIR-NET June 2020. Which of the following protein complexes is involved in licensing of replication origins in eukaryotes? A. CMG complex. B. ORC CDC6 CDT1 MCMC PCNA RFC complex D. cohesin. Answer: B. ORC CDC6 CDT1 MCM. CSIR-NET December 2019. Which of the following is correct regarding origin of replication in budding yeast? A. Contains ORIC-like conserved sequences. B. Contains autonomously replicating sequence Rs C. DNA polymerase initiates replication at Rs D. Rs is GC rich. Answer: B. Contains autonomously replicating sequence ARS. Final thoughts. A comprehensive understanding of replication origin architecture, selection, and control is indispensable for cracking the CSIR-NET JRF Life Sciences exam. This topic sits at the intersection of molecular biology, chromatin dynamics, and cell cycle regulation, making it both conceptually rich and frequently tested. Remember to revise protein components of the pre-replication complex, the licensing mechanism, and the differences between prokaryotic and eukaryotic systems. Mastery of these details will significantly enhance your performance in this high-yield area. You said, replication fork. ChatGPT said. Fundamental processes, replication fork. CSIR-NET JRF Life Sciences level, maximum detail, paragraph format, no tables, no wasted space, most probable topic. The replication fork is a dynamic, Y-shaped structure that forms when DNA unwinds during replication. It is the active site of DNA synthesis where parental DNA strands are separated to allow synthesis of new complementary strands. The fork progresses bidirectionally from each origin of replication, and its coordination is crucial for high-fidelity genome duplication. The architecture and function of the replication fork are conserved across prokaryotes and eukaryotes, though the complexity of regulation differs. At the fork, DNA helicase unwinds the double helix, breaking hydrogen bonds between complementary bases, generating two single-stranded DNA ssDNA templates. These single strands are inherently unstable and prone to forming secondary structures or being degraded. Therefore, single-strand binding proteins SSPs in prokaryotes or replication protein A RPA in eukaryotes bind and stabilize the ssDNA, preventing reannealing or nuclease digestion. The leading strand is synthesized continuously in the 5' to 3' direction, following the direction of fork movement. This synthesis is catalyzed by DNA polymerase III in prokaryotes or DNA polymerase epsilon in eukaryotes. In contrast, the lagging strand is synthesized discontinuously as Okazaki fragments, because DNA polymerases can only add nucleotides in the 5' to 3' direction. These short fragments are later joined by DNA ligase. The enzyme primase lays down RNA primers to initiate synthesis for both strands, once for the leading strand and multiple times for the lagging strand. Helicase, loaded onto the lagging strand template, is responsible for the unwinding process. In E. coli, this is performed by DNA B helicase, while in eukaryotes, the CMG complex CDC45-MCM2 7 gens performs this role. As helicase progresses, it creates topological stress ahead of the fork tube winding of DNA. This is relieved by topoisomerases, particularly DNA gyrase in prokaryotes and topoisomerase I2 in eukaryotes, which prevent fork stalling and DNA breakage. Fork progression is tightly regulated. In eukaryotes, checkpoint kinases like ATR monitor replication stress and can pause fork progression in response to DNA damage or nucleotide depletion. The replication fork protection complex stabilizes the fork and ensures resumption after stalling. Misregulation can lead to fork collapse, genomic instability, and is linked to diseases such as cancer. A key concept is the trombone model of replication. As the lagging strand loops out, the polymerase remains associated with the fork, enabling rapid primer synthesis and fragment elongation. Coordination of leading and lagging strand synthesis is achieved through the replosome complex, which includes helicase, primase, polymerases, and clamp proteins. In prokaryotes, the beta clamp helps tether polymerase to DNA for processivity, while in eukaryotes, this role is played by pCNA proliferating cell nuclear antigen. During S phase, multiple replication forks operate simultaneously in eukaryotic chromosomes. The efficient firing of origins and controlled fork progression are necessary to complete genome replication within a limited time. The replication fork also interacts with chromatin. Histones are displaced and rapidly reassembled onto nascent DNA by histone chaperones, like CAF1 and ASF1, maintaining epigenetic marks through cell division. Fork stalling due to DNA damage, e.g., UV lesions, ROS activates replication stress responses, involving fork reversal or template switching to bypass lesions. Proteins like BRCA1/2, RAD51, and FANCD2 help stabilize and restart stalled forks. Fork reversal involves annealing of nascent strands and reannealing of parental strands, forming a chicken foot structure, which is resolved before replication can resume. CSIRnet previous year question CSIRnet December 2022. Which one of the following statements about the lagging strand synthesis at the replication fork is correct? A. Synthesized continuously by DNA polymerase. B. Synthesized in the 3' to 5' direction. C. Synthesized discontinuously using multiple RNA primers. D. Synthesized in absence of DNA ligase. Answer. C. Synthesized discontinuously using multiple RNA primers. CSIR June 2020. In the eukaryotic replication fork, which complex acts as a replicative helicase? A. pCNA. B. CMG complex. C. Or complex. D. RFC complex. Answer. B. CMG complex. CSIRnet December 2018. In DNA replication, the removal of positive supercoils ahead of the replication fork is carried out by which enzyme? A. Ligase. B. Topoisomerase. C. Primase. D. Helicase. Answer. B. Topoisomerase. Final insights. The replication fork is the central execution unit of DNA replication. Its progression and stability are critical to maintain genome integrity. Understanding the enzymes involved, the molecular mechanics of leading, lagging strand synthesis, fork stalling, and recovery mechanisms is not only key to scoring high in CSIRnet but also fundamental to cell biology. Be thorough with the coordinated roles of polymerases, helicases, primases, clamps, and topoisomerases, and revise models like the trombone loop and fork reversal. This is a highly testable area, and mastering it ensures a strong foundation in molecular biology. You said. Fidelity of replication. ChatGPT said. Fundamental processes, fidelity of DNA replication. CSIRnet JRF Life Sciences level, most probable area, maximum detail, paragraph format, no tables with previous year questions and answers. The fidelity of DNA replication refers to the accuracy with which the DNA polymerases copy the genetic material during cell division. This high fidelity is essential to preserve the integrity of genetic information and prevent mutations that may lead to disease like cancer. Replication fidelity is ensured by the intrinsic selectivity of DNA polymerases, proofreading mechanisms, and post-replicative mismatch repair (MMR) systems. In both prokaryotes and eukaryotes, the error rate of DNA replication is astonishingly low, approximately 1 error per 10 to the power of 9 to 10 nucleotides, despite the massive size of genomes and rapid rate of synthesis. The first layer of fidelity lies in the base pairing specificity guided by Watson-Crick base pairing. DNA polymerases possess a steric gate in the active site that only allows proper purine pyrimidine pairing, minimizing incorporation of incorrect nucleotides. However, this initial selection has a raw error rate of around 1 in 10 to the power of 4 to 10 to the power of 5 bases. To further enhance fidelity, many DNA polymerases, especially DNA polymerase III in prokaryotes and DNA polymerase delta and epsilon in eukaryotes, have intrinsic 3' right pointing arrow 5' exonuclease activity, which functions as a proofreading mechanism. When a mismatched nucleotide is added, the polymerase stalls due to destabilized geometry. The exonuclease then excises the incorrect base, allowing the polymerase to insert the correct nucleotide. The proofreading function significantly improves fidelity by another 100-fold, reducing the error rate to about 1 in 10 to the power of 6 to 10 to the power of 7 bases. Yet, occasional errors still escape this layer. To address these, cells employ the mismatch repair (MMR) pathway, a post-replication correction mechanism that detects and repairs base-based mismatches and insertion-deletion loops. In E. coli, the MMR system includes MUTS, MUTL, and MOVE, which recognize the mismatch, identify the newly synthesized strand based on methylation patterns, and excise the incorrect segment. In eukaryotes, MSH2, MSH6 MUTS homologs, and MLH1P-MS2 MUTL homologs perform analogous functions. The MMR system adds another 100 to 1,000-fold improvement, achieving the ultra-high fidelity observed in vivo. Importantly, replicative DNA polymerases differ from translesion polymerases, which lack proofreading ability and are used during replication across damaged DNA. While translesion synthesis ensures survival under genotoxic stress, it increases mutation rates, emphasizing the role of replicative polymerases in fidelity. Additionally, DNTP balance, divalent cation availability, and chromatin context also influence polymerase accuracy. Eukaryotic polymerases operate in a highly coordinated replosome, where fidelity is also aided by processivity factors like pCNA, which ensure tight interaction with DNA. Furthermore, checkpoint pathways e.g., ATR, CHK1 monitor replication stress and can pause the cycle to allow correction. Defects in DNA polymerase proofreading e.g., pol mutations or MMR e.g., in Lynch syndrome result in hypermutated phenotypes and genome instability. Fidelity is evolutionarily conserved and critical for mutation rate regulation. While high fidelity is essential in somatic cells for stability, a small, regulated mutation rate is beneficial in germ cells and microbial populations to allow evolution and adaptation. CSIRnet previous year question CSIRnet December 2023. Which of the following contributes significantly to the high fidelity of DNA replication? Exonuclease activity. The fidelity of DNA replication refers to the accuracy with which the DNA polymerases copy the genetic material in cell division. This high fidelity is essential to preserve the integrity of genetic information and prevent mutations that may lead to diseases like cancer. The fidelity of DNA replication refers to the accuracy with which the DNA polymerases copy the genetic material during cell division. This high fidelity is essential to preserve the integrity of genetic information and prevent mutations that may lead to diseases like cancer. Replication fidelity is ensured by the intrinsic selectivity of DNA polymerases, proofreading mechanisms, and post-replicative mismatch repair (MMR) systems. In both prokaryotes and eukaryotes, the error rate of DNA replication is astonishingly low, approximately 1 error per 10 to the power of 9 to 10 nucleotides, despite the massive size of genomes and rapid rate of synthesis. The first layer of fidelity lies in the base pairing specificity guided by Watson-Crick base pairing. DNA polymerases possess a steric gate in the active site that only allows proper purine pyrimidine pairing, minimizing incorporation of incorrect nucleotides. However, this initial selection has a raw error rate of around 1 in 10 to the power of 4 to 10 to the power of 5 bases. To further enhance fidelity, many DNA polymerases, especially DNA polymerase III in prokaryotes and DNA polymerase delta and epsilon in eukaryotes, have intrinsic 3' right pointing arrow 5' exonuclease activity, which functions as a proofreading mechanism. When a mismatched nucleotide is added, the polymerase stalls due to destabilized geometry. The exonuclease then excises the incorrect base, allowing the polymerase to insert the correct nucleotide. The proofreading function significantly improves fidelity by another 100-fold, reducing the error rate to about 1 in 10 to the power of 6 to 10 to the power of 7 bases. Yet, occasional errors still escape this layer. To address these, cells employ the mismatch repair (MMR) pathway, a post-replication correction mechanism that detects and repairs base-based mismatches and insertion-deletion loops. In E. coli, the MMR system includes MUTS, mutyl, and MOVE, which recognize the mismatch, identify the newly synthesized strand based on methylation patterns, and excise the incorrect segment. In eukaryotes, MSH2, MSH6 MUTS homologs and MLH1P-MS2 mutyl homologs perform analogous functions. The MMR system adds another 100 to 1,000-fold improvement, achieving the ultra-high fidelity observed in vivo. Importantly, replicative DNA polymerases differ from translesion polymerases, which lack proofreading ability and are used during replication across damaged DNA. While translesion synthesis ensures survival under genotoxic stress, it increases mutation rates, emphasizing the role of replicative polymerases in fidelity. Additionally, DNTP balance, divalent cation availability, and chromatin context also influence polymerase accuracy. Eukaryotic polymerases operate in a highly coordinated replosome, where fidelity is also aided by processivity factors like pCNA, which ensure tight interaction with DNA. Furthermore, checkpoint pathways e.g., ATR, CHK1 monitor replication stress and can pause a cycle to allow correction. Defects in DNA polymerase proofreading e.g., pol mutations or MMR e.g., in Lynch syndrome result in hypermutated phenotypes and genome instability. Fidelity is evolutionarily conserved and critical for mutation rate regulation. While high fidelity is essential in somatic cells for stability, a small, regulated mutation rate is beneficial in germ cells and microbial populations to allow evolution and adaptation. CSIRNet previous year question CSIRNet December 2023. Which of the following contributes significantly to the high fidelity of DNA replication? Exonuclease activity of DNA helicase. B 3' to 5' exonuclease activity of DNA polymerase C, 5' to 3' polymerase activity of topoisomerase D, action of DNA primase. Answer: B 3' to 5' exonuclease activity of DNA polymerase. CSIRNet June 2019. In mismatch repair in prokaryotes, how is the newly synthesized strand identified for correction? A based on ATP hydrolysis. B by RNA primer presence, C by lack of methylation, D by Okazaki fragments. Answer: C by lack of methylation. CSIRNet Deck 2017. What would be the likely consequence of a mutation that inactivates the proofreading activity of DNA polymerase? A reduced replication speed. B. Increased DNA damage detection. C. Increased mutation rate. D. Failure of Okazaki fragment ligation. Answer: C. Increased mutation rate. Final insights. The fidelity of DNA replication is a multi-layered defense system that ensures the faithful transmission of genetic information. It integrates base selectivity, proofreading, and post-replicative MMR mechanisms. Understanding the enzymes, checkpoints, and mutations affecting this process is crucial not only for CSIRNet success but also for broader insights into cancer biology, microbial adaptation, and genetic diseases. As fidelity mechanisms are frequently tested in CSIR exams, mastering their molecular bases and applications is essential. You said. Extrachromosomal replicons. ChatGPT said. Fundamental processes. Extrachromosomal replicons. CSIRNet JRF Life Sciences Level Full Length High Yield Lecture. Paragraph format. Maximum content with previous year questions and answers. Extrachromosomal replicons are DNA molecules that replicate independently of the chromosomal DNA within a cell. These replicons possess their own origin of replication and essential replication machinery, allowing them to maintain and propagate themselves autonomously. They play significant roles in microbial physiology, horizontal gene transfer, genetic variation, and biotechnology. The term replicon refers to any unit of DNA capable of replicating from a single origin of replication, and extrachromosomal replicons are those which exist outside the main chromosomal framework of the cell. Common examples include plasmids, bacteriophages, episomes, mitochondrial and chloroplast DNA, and certain viruses. Plasmids are the most widely studied extrachromosomal replicons, especially in prokaryotes like Escherichia coli. They are typically circular, double-stranded DNA molecules, ranging in size from a few kilobases to hundreds of kilobases. Plasmids carry non-essential genes, but these often provide selective advantages such as antibiotic resistance, virulence factors, or metabolic capabilities. Replication of plasmids may be beta-type, similar to chromosomal replication, or rolling circle, commonly seen in small plasmids in certain phages. Plasmid replication is regulated by the copy number control mechanisms, which ensure that plasmid burden is minimized in the host. Some plasmids, known as conjugated plasmids, like the F plasmid in E. coli, encode the machinery required for horizontal gene transfer via bacterial conjugation. Others, called non-conjugated plasmids, require assistance for transfer. Plasmids may also exist as episomes, which are capable of integrating into the host chromosome, as seen with the after in E. coli. Integration and excision from the chromosome are tightly regulated and involve site-specific recombination. In eukaryotes, extrachromosomal replicons are found in organelles such as mitochondria and chloroplasts, which contain their own circular DNA molecules. Mitochondrial DNA, mtDNA, and chloroplast DNA, cpDNA, replicate independently of the nuclear genome, using replication machinery that shares similarity with prokaryotic enzymes, supporting the endosymbiotic theory. Replication of organelle DNA is semi-conservative and is regulated by specific organelle-localized DNA polymerases, e.g., DNA polymerase gamma in mitochondria. Another important class includes bacteriophages, which can exist as extrachromosomal elements in their lysogenic form. The lambda phage of E. coli, for instance, can replicate autonomously or integrate into the host chromosome. Certain animal viruses, such as Epstein-Barr virus (EBV), maintain their genomes as extrachromosomal replicons in the nuclei of infected cells, contributing to persistent infections and even oncogenesis. Additionally, extrachromosomal circular DNAs (ECCNAs) or double minutes are sometimes observed in cancer cells, where they can carry amplified oncogenes and contribute to genetic heterogeneity and drug resistance. These elements replicate independently and are not part of the chromosomal structure, offering potential diagnostic and therapeutic targets. Extrachromosomal replicons are exploited in molecular biology and genetic engineering. Artificial plasmids engineered with selectable markers, origin of replication ORI, and multiple cloning sites are fundamental tools in recombinant DNA technology, gene cloning, and expression studies. The origin must be compatible with the host cell's replication system, and modifications allow control over copy number, expression level, and inducibility. Understanding the behavior, structure, and regulation of extrachromosomal replicons is crucial in both basic and applied biological sciences. Their ability to move between cells, carry adaptive genes, and act independently from the host genome makes them central to evolutionary dynamics and biotechnological innovations. CSIRNet previous year question CSIRNet December 2022. Which of the following is a key feature of plasmid DNA replication that differentiates it from chromosomal replication in bacteria? A bidirectional replication only. B. Origin of replication dependent on ORIC. C. Autonomous replication independent of the host chromosome. D. Use of RNA primers. Answer, C. Autonomous replication independent of the host chromosome. CSIR Net June 2020. Which one of the following statements about episomes is true? A. They are always linear DNA. B. They cannot integrate into host chromosome. C. They are plasmids that can exist both integrated and independent of the host chromosome. D. They encode rRNA genes. Answer, C. They are plasmids that can exist both integrated and independent of the host chromosome. CSIR Net Deck 2017. A plasmid with a high copy number is most likely to A. Be found only in eukaryotic cells. B. Express more of a clone gene product. C. Be unable to replicate autonomously. D. Require integration into the host genome for maintenance. Answer, B. Express more of a clone gene product. Final notes. Extrachromosomal replicons are an essential concept frequently tested in CSIR Net Sciences. Questions often focus on their structure, replication, evolutionary role, and application in biotechnology. A deep understanding of how these DNA elements function independently and influence host physiology is critical for mastering microbial genetics and molecular biology, both core domains in the CSIR Net syllabus. You said, DNA damage. ChatGPT said. Fundamental processes, DNA damage. CSIR Net JRF Life Sciences based to advanced level full-length high-yield lecture paragraph format maximum content with PYQs. DNA damage refers to alterations in the chemical structure of DNA that compromise its integrity and potentially its ability to replicate or express information accurately. It is distinct from mutation. While mutations are heritable sequence changes, DNA damage includes physical or chemical abnormalities that may be repaired before becoming permanent. Cells continuously experience endogenous damage due to metabolic processes and exogenous damage from environmental agents. If unrepaired or misrepaired, such lesions can result in mutations, genomic instability, or cell death, contributing to cancer, aging, and genetic diseases. Types of DNA damage include single base modifications, strand breaks, crosslinks, and bulky adducts. The most common endogenous source of DNA damage is reactive oxygen species, ROS, generated during oxidative phosphorylation. ROS can cause base oxidation, strand breaks, and basic sites. One of the most studied oxidative lesions is 8-oxodeoxyguanosine, 8-oxo-DG, which can misfire with adenine, causing GC to TOT transversions. Depurination and deamination are spontaneous chemical events leading to a pyrimidinic AP sites or base substitutions. Deamination of cytosine produces uracil, which pairs with adenine during replication, resulting in transition mutations if unrepaired. Replication errors, though corrected by proofreading and mismatch repair, may also lead to misincorporated bases. Exogenous DNA damage agents include ultraviolet UV radiation, which induces the formation of pyrimidine dimers, especially thymine dimers. These distort the DNA helix and block replication and transcription. Ionizing radiation, X-rays, gamma rays, which causes single and double strand breaks, SSBs and DSBs, base damage, and crosslinking. Chemical mutagens like alkylating agents, e.g., AMPs, intercalating agents, e.g., ethylene bromide, and crosslinkers, e.g., cisplatin modify base pairing properties, create bulky adducts, or interfere with DNA topology. Double strand breaks, DSBs, are among the most lethal types of damage. They can be repaired by homologous recombination, HR, or non-homologous end joining, NHEJ. Improper repair of DSBs can lead to chromosomal rearrangements, deletions, or oncogenic transformations. To combat these threats, cells employ a range of DNA repair mechanisms, each specialized for particular types of lesions. For example, base excision repair, BER, targets small, non-helix distorting base lesions such as 8-oxo-DG or uracil. The damaged base is removed by DNA glycosylase, followed by end processing, gap filling by DNA polymerase, and ligation. Nucleotide excision repair, NER, removes bulky lesions like UV-induced pyrimidine dimers. The defect is excised as an oligonucleotide, and the gap is filled using the undamaged strand as a template. Mismatch repair, MMR, corrects base mismatches and insertion deletion loops generated during replication. Defects in MMR are linked to hereditary non-polyposis colorectal cancer, HNPCC. Homologous recombination, HAUR, is an error-free mechanism using a sister chromatid as a template, active in SG2 phases. Non-homologous end joining, NHEJ, rejoins broken DNA ends without sequence homology, often leading to small insertions or deletions. DNA damage response, DDR, is a complex signaling network that detects damage, halts cell cycle progression, and activates repair or apoptosis. Central to DDR is the ATM-ATR kinase pathway. ATM-ataxia-telangiectasia mutated is activated by DSBs and phosphorylates downstream targets like P53 and CHK2. ATR responds to single-stranded DNA at stalled replication forks and activates CHK1. P53, a tumor suppressor, arrests the cell cycle at G1S by inducing P21 or triggers apoptosis if damage is irreparable. Mutations in P53 are implicated in over 50% of human cancers. Some cells, upon excessive DNA damage, undergo senescence or programmed cell death, both of which are crucial to prevent the proliferation of genetically compromised cells. DNA damage-induced senescence is a permanent cell cycle arrest marked by changes in chromatin and gene expression profiles. In biotechnology and medicine, DNA damage is exploited in cancer therapies like radiation and chemotherapy, which induce DNA breaks in rapidly dividing cells. However, tumor cells may develop resistance by enhancing DNA repair mechanisms, an active area of cancer research. Understanding DNA damage is also critical in genome editing, aging research, and regenerative medicine. CSIR Net previous year questions, PYQs, CSIR Net December 2019. Which of the following DNA lesions is primarily repaired by nucleotide excision repair, NER? A. Uracil incorporation. B. Thymine dimer. C. 8-oxo-guanine D. Mismatched basis. Answer, B. Thymine dimer. CSIR Net June 2017. Which one of the following types of DNA damage results from reactive oxygen species, ROS? A. Crosslinking of DNA strands. B. Deamination of cytosine. C. Formation of thymine dimers. D. Oxidation of guanine to 8-oxo-DG. Answer, D. Oxidation of guanine to 8-oxo-DG. CSIR Net December 2016. Defects in mismatch repair genes are most commonly associated with A. Xeroderma pigmentosum. B. Ataxia-telangiectasia. C. Hereditary non-polyposis colorectal cancer, HNPCC. D. Bloom syndrome. Answer, C. Hereditary non-polyposis colorectal cancer, HNPCC. This lecture delivers all you need to master DNA damage for CSIR Net JRF. Let me know when you're ready for the next topic. You said, repair mechanisms, ChatGPT said. Fundamental processes, DNA repair mechanisms. CSIR Net JRF Life Sciences based to advanced ultra-detailed high-yield theory, PYQs included paragraph format. DNA repair mechanisms are essential cellular processes that preserve genome integrity by correcting damage caused by endogenous metabolic byproducts or exogenous mutagens like UV light, ionizing radiation, and chemicals. Since DNA is the permanent repository of genetic information, even minor damage can lead to mutations, genomic instability, apoptosis, or cancer if not promptly and accurately repaired. Cells employ a range of highly conserved and specialized repair pathways tailored to specific types of DNA lesions. These mechanisms are not only fundamental to cell survival but are also crucial during replication, recombination, and development. The major DNA repair systems include direct repair, base excision repair, BER, nucleotide excision repair, NER, mismatch repair, MMR, homologous recombination, HR, and non-homologous end joining, NHEJ. 1. Direct reversal of damage is the simplest form of repair and involves the enzymatic removal of chemical modifications without breaking the DNA backbone. One prominent example is the repair of O6-methylguanine by O6-methylguanine DNA methyltransferase, MGMT, which transfers the methyl group to a cysteine residue in its active site, restoring guanine and rendering the enzyme inactive. Similarly, DNA photolysis, present in prokaryotes and lower eukaryotes, use visible light energy to split UV-induced cyclobutane pyrimidine dimers, CPDs, in a process called photoreactivation. Similarly, DNA photolysis, present in the major DNA repair systems include direct repair, base excision repair (BER), nucleotide excision repair (NER), mismatch repair (MMR), homologous recombination (HR), and non-homologous end joining (NHEJ). The major DNA repair systems include direct repair, base excision repair (BER), nucleotide excision repair (NER), mismatch repair (MMR), homologous recombination (HR), and non-homologous end joining (NHEJ). 1. Direct reversal of damage is the simplest form of repair and involves the enzymatic removal of chemical modifications without breaking the DNA backbone. One prominent example is the repair of O6-methylguanine by O6-methylguanine DNA methyltransferase (MGMT), which transfers the methyl group to a cysteine residue in its active site, restoring guanine and rendering the enzyme inactive. Similarly, DNA photolysis, present in prokaryotes and lower eukaryotes, use visible light energy to split UV-induced cyclobutene pyrimidine dimers (CPDs) in a process called photoreactivation. However, humans lack photolysis and rely on NER for such lesions. 2. Base excision repair (BER) targets small, non-helix distorting base lesions like deaminated, oxidized, or alkylated bases. The repair is initiated by DNA glycosylases, which recognize and remove the damaged base, leaving in a basic AP site. An AP endonuclease then cleaves the DNA backbone at the AP site, followed by DNA polymerase beta-mediated gap filling and DNA ligase sealing. Short patch BE replaces a single nucleotide, while long patch BER can replace 2 to 10 nucleotides, especially during replication or oxidative stress. 3. Nucleotide excision repair (NER) removes bulky, helix-distorting lesions like thymine dimers and chemical adducts. It operates via 2 sub-pathways: global genomic NER (GGNER) that surveys the whole genome, and transcription-coupled NER (TCNER) that targets lesions blocking transcription. The damage is recognized by XPC in GGNER or CSA/CSB in TCNER, which recruit the TFIIH complex. XPB and XPD helicases unwind the DNA, and XPFERCC1 and XPG endonucleases excise approximately 24 to 32 nucleotide fragment. DNA polymerase fills the gap using the undamaged strand as a template, and ligation comes the repair. Mutations in NER genes cause disorders like xeroderma pigmentosum and Cockayne syndrome. 4. Mismatch repair (MMR) corrects replication errors such as base-base mismatches and insertion-deletion loops. In prokaryotes, MUTS recognizes a mismatch, mutyl recruits MUT, which introduces a nick in the newly synthesized strand identified by its lack of methylation. The segment containing the mismatch is then excised by exonucleases, and the gap is filled and ligated. In eukaryotes, MSH2, MSH6, MUTS alpha, and MLH1, PMS2, mutyl alpha are the main homologs. Defects in MMR are linked to hereditary non-polyposis colorectal cancer (HNPCC) or Lynch syndrome, underlining its clinical importance. 5. Double-strand break repair (DSBR) is critical for survival, as DSBs are among the most lethal DNA lesions. There are 2 major DSBR pathways: homologous recombination (HR) and non-homologous end joining (NHEJ). Homologous recombination (OR) is an error-free mechanism active during S and G2 phases when a sister chromatid is available. The DSB ends are processed to form 3' single-stranded overhangs, which invade the homologous template via the Rad51 recombinase, forming a holiday junction. DNA synthesis extends the invading strand, followed by resolution and ligation. HR is vital for post-replicative repair and meiosis. Non-homologous end joining (NHEJ) is error-prone as it directly ligates broken ends without using a homologous template. Key components include the Cu70, Cu80 heterodimer, DNA PKCs, Artemis for end processing XRCC4, and DNA IB. Though fast, it can cause small insertions or deletions indels and is more prone to chromosomal translocations, often contributing to tumorigenesis if deregulated. 6. Translesion DNA synthesis (TLS) is a damage tolerance process where specialized Y family DNA polymerases, like POL-eta, POL-iota, POL-kappa replicate across damaged templates that would otherwise stall the replication fork. Though inherently error-prone, TLS allows continuation of replication. POL-eta is specifically known for bypassing UV-induced thymine dimers, and its deficiency causes XP variant phenotype. DNA damage response (DDR) acts as a surveillance mechanism, activating cell cycle checkpoints via ATM, ataxia-telangiectasia mutated, and ATR, ATM, and RAD3 related kinases. Upon DSBs, ATM phosphorylates CHK2, BRCA1, and P53, leading to cell cycle arrest, repair, or apoptosis. ATR responds to replication stress and single-stranded DNA via CHK1 activation. P53, the guardian of the genome, decides cell fate depending on the extent of damage. Defective DDR is central to many cancers and neurodegenerative diseases, and targeting DNA repair proteins, e.g., PARP inhibitors in BRCA-mutated cancers, is a therapeutic strategy. In conclusion, DNA repair is not a redundant backup but an integrated, highly regulated system central to life. From maintaining cellular fidelity to determining cancer susceptibility, these pathways highlight how biological systems have evolved to guard against inevitable molecular imperfections. CSIRnet previous year questions PYQs CSIRnet December 2022. Which of the following enzymes is involved in the direct repair of O6-methylguanine in DNA? A DNA polymerase B. Uracil DNA glycosylase C. O6-methylguanine DNA methyltransferase D. Exonuclease I. Answer: C. O6-methylguanine DNA methyltransferase. CSIRnet June 2015. Mismatch repair in humans is primarily mediated by which of the following heterodimers? A. Rad50 MRE11. B. MSH2 MSH6 C. BRCA1 BRCA2 D. ATM ATR. Answer: B. MSH2 MSH6. CSIRnet December 2016. A cell deficient in nucleotide excision repair is likely to be hypersensitive to a gamma radiation. B. UV radiation C. Oxidative stress D. Ionizing radiation. Answer: B. UV radiation. Let me know if you want the next topic in this series, such as homologous recombination, gene expression, or any other CSIRnet unit. You said. Homologous and site-specific recombination. ChatGPT said. Fundamental processes. Homologous and site-specific recombination. CSIRnet JRF Life Sciences. Ultra-detailed, exam-oriented theory based to advanced PYQs included paragraph format. Recombination is a fundamental process in all domains of life, crucial for maintaining genetic integrity, generating diversity, and facilitating proper chromosomal segregation during meiosis. 2 major types of recombination are extensively studied and tested in CSIRnet Life Sciences: homologous recombination (HR) and site-specific recombination (SSR). Each operates through precise molecular mechanisms involving specific enzymes and recognition sequences, and the regulation is essential for genome stability, development, and evolution. Homologous recombination (HR). Homologous recombination is a conserved, error-free DNA repair and recombination mechanism that involves the exchange of genetic material between 2 homologous DNA molecules. This process is pivotal for meiotic chromosomal crossover, post-replicative DNA repair, and the reactivation of stalled replication forks. HR is especially active in the S and G2 phases of the cell cycle when a sister chromatid is available. The process initiates when a double-strand break (DSB) occurs, often due to replication fork collapse or programmed events during meiosis. The break is recognized by the MRN complex MRE11 Rad50 NBs1, which processes the DNA ends via 5' to 3' resection, producing 3' single-stranded overhangs. These overhangs are immediately coded by RPA, replication protein A, to prevent secondary structures. Subsequently, Rad51, a recombinant homologous to bacterial RECA, replaces RPA and forms a nucleoprotein filament that invades the homologous double-stranded DNA on the sister chromatid or homologous chromosome, forming a D-loop displacement loop. Once strand invasion is complete, DNA polymerase extends the 3' end using the homologous sequence as a template. This can be resolved by several mechanisms. Synthesis-dependent strand annealing (SDSA), where the invading strand dissociates and anneals to the other processed end, resulting in non-crossover products. Double holiday junction (DHJ) formation, which can be resolved by resolvases like Gen1 to produce either crossover or non-crossover products, depending on the orientation of cleavage. Break-induced replication BER, an extensive recombination event used when only one end of a DSB has homology, typically observed in telomere maintenance or collapsed forks. HR also plays a crucial role in meiosis I, where recombination between homologous chromosomes ensures proper alignment and segregation. Proteins such as SPO11 initiates meiotic DSBs, RAD52, and DMC1 meiosis-specific recombinase are central to meiotic hour. In eukaryotes, hour is tightly regulated by cell cycle checkpoints and is suppressed in G1 to avoid recombination with non-sister chromatids, which may lead to chromosomal aberrations. Site-specific recombination (SSR). In contrast to HR, site-specific recombination involves the rearrangement of DNA segments at specific short sequences 20 to 200 BP recognized by recombinase enzymes. SSR is not dependent on extensive sequence homology and is often used for integration, excision, inversion, or translocation of DNA elements such as bacteriophage genomes or mobile genetic elements. SSR is widely used in genetic engineering, e.g., pre-LOXP and FLP-FRT systems. There are two major types of recombinases involved in SSR. Tyrosine recombinases, e.g., pre-FLP, lambda integrase, they act through a holiday junction intermediate, cleaving and rejoining DNA one strand at a time. The process involves the formation of a holiday junction followed by isomerization and strand exchange. Lambda phage integrase is a classic example, which mediates integration of lambda DNA into the E. coli genome at the ATTP and ATTB sites. Serine recombinases, e.g., resolvase, HIN invertase, these recombinases simultaneously cut all 4 DNA strands across the recombination sites and swap them without forming a holiday junction. This mechanism is faster and more suited for large-scale DNA rearrangements, like inversion and transposition. SSR is also used by bacteria and viruses to evade host immunity via phase variation, and by the immune system in higher eukaryotes for VTJ recombination, which generates the vast diversity of antigen receptors. RAG1 and RAG2 recombinases, along with non-homologous end-joining NATJ proteins, mediate this specialized SSR in lymphocytes. In molecular biology, SSR systems like pre-LOXP have revolutionized conditional gene expression, allowing tissue-specific and temporally controlled gene knockout or activation in model organisms such as mice. Distinguishing HR and SSR. While both processes involve recombination, HR requires long stretches of sequence similarity and functions primarily in DNA repair and meiosis, whereas SSR targets specific recombination sequences and is often used for DNA rearrangement, integration, or gene regulation. HR is more versatile in maintaining genomic integrity, while SSR is crucial for precise rearrangements in genetic circuits. CSIR-NET previous year questions PYQs CSIR-NET June 2014. Homologous recombination requires which of the following proteins in eukaryotes? BRCA. B RAD51C RAG1D integrase answer B RAD51. CSIR-NET December 2016. Which of the following statements is true about site-specific recombination? It requires long stretches of homology. B. It proceeds through holiday junctions only. C. It occurs at specific DNA sequences. D. It does not involve DNA cleavage answer. C. It occurs at specific DNA sequences. CSIR-NET June 17. Pre-recombinase recognizes which of the following sequences? FRT. B. ATTBC LOXP D. ORIC answer C. LOXP.